Jesus said, for judgment, I've come into, oh gosh, that's not picking me up. Let's try this. Jesus said, for judgment, I come into the world. Okay, yes, that works. Okay. Jesus said, for judgment, I've come into the world. All right, gang, it's time to start. I finished the urinary system. I did a little podcast using PowerPoint. It is online on Blackboard. So go ahead and avail yourself to that. So we're going to start the reproductive system. Today we're starting with the male reproductive system because it's simpler. Not because I'm a sexist pig or something like that, but we're going for simple first. Then we'll do the female one that's a bit more complicated. And then we'll go from there. And when I don't finish, I will podcast. All right, so without further ado, let's begin. Father, thank you for this day. We pray we get a bit more sun. Thank you for this cold weekend we're apparently going to have. We pray as we see the end in sight, you give us a nice kick towards the end. Give us a boost. We're not in class. We don't see each other's smiling faces. But help us, Lord, to have a smile inside, knowing we belong to you. In Jesus' name, amen. So the male reproductive system consists of internal and external organs and structures that are about the process of reproduction. The internal organs, of course, include the testes. These are the site of sperm production, and they are the male gonads. They are suspended in a sac known as the scrotum. Other internal glands include so-called accessory sex glands that make secretions that are put into the mixture of sperm that produces a mixture known as semen. So the scrotum hangs outside the body. It has several layers of muscle and fascia that keep the testes temperature about 3 degrees lower than the body temperature. That is a prime temperature for sperm production. When it's cold outside, the scrotal skin will wrinkle and it will elevate and bring the testes closer to the body to warm up. And when it's warm outside, the scrotal skin will loosen up and the scrotum will lower the testes away from the body and heat will be released through sweating. The testes themselves are covered with a white fibrous layer called the tunica albuginea. Tunica albuginea. That's A-L-B-U-G-I-N-E-A. A-L-B-U-G-I-N-E-A. Tunica albuginea. So a optical section through a testes shows that we have these fibrous projections. You can see them here. These are called testal septa. These move towards the center of the testis. The septa partition each testis into 250 lobules. Each lobule will have up to at least 1 and up to 4 of these seminiferous tubules. And the seminiferous tubule is the site where sperm are produced. The seminiferous tubules connect to a single straight tubule that exits this lobule and enters a network called the rete testes. The rete testes. The rete testes, as you can see, are small networks of ducts that split up, come together again, kind of like catacombs. And located in the center of the testis, right here, is the so-called mediastinum testes. And then the sperm make it through the rete testes and go through the efferent ductules to the epididymis. The epididymis is a comma-shaped organ. It has a head and a tail. And we're going to zoom in on these seminiferous tubules now. So the seminiferous tubule has on the outside a thick wall of epithelial cells. They surround a fluid-filled lumen. Outside the testis is a network of capillaries, also embedded in connective tissue. And there are cells on the top also called Leydig cells. And these are the cells that produce testosterone. So here is a dog testis. Structure is the same. You can see the lobules of the testis. You can see the septa of the tunica albuginea. You can see the tunica albuginea here. And this outer layer is called the tunica vaginalis. And these then flow into the rete testis, into the mediastinum. Here's the epididymis. So if we take a look at a close-up of a seminiferous tubule here, the wall of the seminiferous tubule is composed of 3 kinds of cells. At the periphery, you have the spermatogonia. These are the stem cell population that divide over and over again to produce sperm. The products of their division and the spermatogonia, as you can see, are found at the periphery of the seminiferous tubule. So they divide by mitosis. One cell, the cell that remains basal, remains a spermatogonium. The other cell produces a primary spermatocyte. These primary spermatocytes can divide mitotically, but only a few times. So they will, the primary spermatocyte is diploid. It has 23 pairs, chromosome pairs, so 20 pairs of chromatids, so 46 chromatids. They will undergo S phase. They'll then make 46 chromosomes, 92 chromatids, and they will then enter meiosis. As they enter meiosis, they will form secondary spermatocytes, and those secondary spermatocytes will mature into spermatids that then mature into spermatogonia. So essentially you're going, as you go from basal to apical, you become more mature. And here's a good picture of the Leydig cells. On the outside, there's the connective tissue, there's smooth muscle out here as well. And there's the Leydig cells. So the production of sperm, spermatogenesis, begins during puberty and is so under the control of the hypothalamus. The hypothalamus will produce gonadotropin releasing hormone that travels to the anterior lobe of the pituitary, which will then secrete two of its own hormones, the so-called gonadotropins. These gonadotropins consist of follicle stimulating hormone and luteinizing hormone. Before puberty, both gonadotropin releasing hormone, follicle stimulating hormone, and luteinizing hormone are secreted in low, constant amounts. At puberty, the secretion of these hormones becomes pulsatile. Sometimes more is released, sometimes less is released. Luteinizing hormone will bind to receptors on the surfaces of Leydig cells. In response, the Leydig cells will up the production of testosterone. Follicle stimulating hormones will bind to the surface of and stimulate a cell in the seminiferous tubule called the Sertoli cell. And this cell will start to make androgen binding protein, ABP. This allows more testosterone to cross the blood-testis barrier and enter the seminiferous tubule. That really gets spermatogenesis going. The seminiferous tubule will also produce, in particular the Sertoli cell, will produce inhibin in response to follicle stimulating hormone that will inhibit the amount of follicle stimulating hormone, and testosterone will inhibit the amount of luteinizing hormone. That puts a check on the amount of these gonadotropins. Interestingly, inhibin is also made by the ovary. So the levels of these gonadotropins does increase, but it only increases to a certain level. They won't go terrifically high because testosterone is shutting it down, and inhibin, which is induced by testosterone, is shutting it down. And this picture shows you this negative feedback cycle that's so characteristic of endocrine hormones. The brain is also a target for testosterone. Interestingly, in the brain, testosterone is converted to an estrogen, estradiol 17 beta, and this is an important feedback inhibitor of luteinizing hormone secretion by testosterone. And interestingly, in laboratory mice that fail to convert testosterone to estradiol 17 beta do not properly feedback inhibit the production of luteinizing hormone. So the production of estrogen from testosterone is due to the work of an enzyme called aromatase. Testosterone is also converted to 5-alpha dihydrotestosterone by 5-alpha reductase, and then 5-alpha dihydrotestosterone is converted into androstenediol. And these androstenediols are two other androgens that do some of the work of androgens. So gonadotropin production in males remains relatively constant. It does decrease with age, but it decreases gradually. You don't get the precipitous drop in sex steroids like you do in females and menopause. And testosterone and its derivatives, the 5-alpha dihydrotestosterone and androstenediols, they are responsible for the body changes associated with puberty in males. And here are some of those features. And there is a puberty gradation called the Tanner scale that is used to grade how males are going through puberty. And family physicians who take care of young men and young women, they will examine them for progression through the Tanner stage in order to make sure that their growth and development is normal. Because steroids cause growth of organs, growth of the skeleton, growth of skeletal muscle, increase red blood cell production, enlargement of things like the prostate, the testes, the penis, things like that, they're often called anabolic steroids. Males will typically have a lower voice than females because androgens will cause the growth of the larynx, dropping the voice. They also promote hemoglobin production as well. Interestingly, your Sertoli and Leydig cells also contain receptors for estrogen. And male mice that do not have a functional copy of the estrogen receptor are sterile. And why that's the case is not well understood. So, something to keep in mind, your book points this out. You have two compartments, the seminiferous tubules, the extra seminiferous tubule compartment. These two compartments are controlled by two different hormones. One's controlled by LH, the other by FSH, yet these two components talk to each other and regulate each other, and the communication is largely by means of the hormone testosterone. And Leydig cells also produce peptide hormones that were previously thought to only be made by the anterior pituitary. And these include adrenocorticotropic hormone. They don't make a lot of adrenocorticotropic hormone, but they make some. Melanocyte-stimulating hormone and beta endorphin. And ACTH and MSH stimulate Sertoli cell function, and beta endorphin will inhibit Sertoli cell function. Very good. Let's look at spermatogenesis. All right, during early embryonic development, you have the formation of a structure. It's now called the primary umbilical vesicle. It was formerly called the yolk sac. It's not called the yolk sac anymore, although old books and old embryologists will still call it the yolk sac because old habits die hard. In the wall of the primary umbilical vesicle, formerly called the yolk sac, you have groups of cells that start to come out and divide and move by amoeboid movement. These cells are called the primordial germ cells. They migrate up the wall of the primary umbilical vesicle into the developing gut. So they move through the hindgut and they move into the midgut. Then they descend through the midgut into the developing gonad. These cells will become the gametes. So these spermatogonia, again, they're diploid cells. There's initially some 1 to 2,000 of these primordial germ cells that will migrate into the testes. They will divide and form the spermatogonia that line the seminiferous tubules. So they divide to form primary spermatocytes, but those primary spermatocytes will usually not undergo meiosis until puberty. So once again, the division of the spermatogonial cell produces two cells. The cell that is displaced apically becomes the primary spermatocyte. The cell that stays basal remains a spermatogonium. And you can see this here. So follow the arrow. Let me put my pen here. Okay, so we start out with the spermatogonium. So this has 23 pairs of chromosomes. It divides, forms a cell that also has 23 pairs of chromosomes. That will then undergo S phase, and now it will have 46 chromosomes, 92 chromatids. That's a primary spermatocyte. Once it divides, and becomes a primary spermatocyte. It will form a type of primary spermatocyte that can divide mitotically. It only stays in the state for a very short time. If it does not divide mitotically, it will not. And if it does divide mitotically, it only usually divides once. Some of these spermatogonia are not actively dividing. Some of them are quiescent. They don't participate in making sperm unless there's a need for more sperm. So there's kind of a storage population of spermatogonia, and they're given different names. There's a type A dark spermatogonium, a type A pale spermatogonium, and a type B spermatogonium. The type A dark spermatogonium can divide to produce more cells. The dark spermatogonia will typically divide to replenish spermatogonia. The type A pale spermatogonia will divide to form primary spermatocytes. And the type B spermatogonia only divide to produce primary spermatocytes. So this cell then undergoes meiosis I, and it becomes a secondary spermatocyte. And when these cells undergo meiosis, the products are joined still by cytoplasmic bridges. They don't separate completely just yet. Then these secondary spermatocytes undergo meiosis again. And when they undergo meiosis again, they become spermatids. Notice they're still joined. And then they will mature further and become sperm cells. And notice when they become sperm cells, they lose the cytoplasmic connection to each other. That's very important. So the production of sperm from spermatids, that's called spermiogenesis. Spermiogenesis. So spermiogenesis has 4 stages. There's a so-called Golgi stage. There's the cap stage. There's the acrosomal stage. And then there's the maturation stage. During the Golgi stage, the sperm develop polarity. And the sperm, which are kind of bullet-shaped, they have a vesicle at the top full of digestive enzymes called the acrosome. And the acrosome is made by vesicles that bud off the Golgi apparatus. Also during this stage, DNA undergoes exceedingly tight packaging. So the cap stage is called the cap stage because the Golgi surrounds the condensed nucleus and it starts to secrete the acrosome. So the Golgi now essentially forms a cap, and the cap essentially starts to secrete the acrosome. So the Golgi cap starts to secrete the acrosome. And then at the acrosome stage, one of the centrioles now elongates and it forms a structure called the manchette. The manchette. Here's a picture. So here is essentially the elongation. You're starting to make the flagellum, and this is rooted here in this centriole. This elongating structure, that's your manchette. That's what's meant by the manchette. And then during the maturation stage, the sperm is going to dump residual cytoplasm it doesn't need. And those are the stages essentially of spermiogenesis. It's much more complicated than that, but that's the Reader's Digest version. Now, these sperm, as they develop, they're embedded in this large cell called a Sertoli cell. The Sertoli cell surrounds the spermagonium. It also surrounds the primary, secondary spermatids, also the spermatids, and also the developing sperm. And then this residual body that it casts off, that gets phagocytized by the Sertoli cell. This phagocytosis of the residual body is a testosterone-dependent process. When it does that, it releases the sperm into the lumen of the seminiferous tubule. So maturation requires the Sertoli cell. It requires testosterone. And once the sperm are mature, they're released into the lumen of the seminiferous tubule, but they are not yet motile. It's very important to understand that, not yet motile. And to remind you, this whole process requires testosterone. In response to FSH, the Sertoli cell synthesizes androgen-binding protein that concentrates testosterone within the Sertoli cells and within the lumen of the seminiferous tubule. It potentiates and accelerates the production of sperm by the seminiferous tubule. It takes roughly 2 months for spermatogonia to develop into sperm. And this is all a hormone-directed process. After puberty, is completed, males will produce about 100 million sperm per day. That's a lot of sperm. So before puberty, the primary spermatocytes are arrested. Testosterone is necessary for the completion of meiotic divisions. Follicle stimulating hormone is required for the maturation of spermatids into spermatozoa that are released by the Sertoli cell. Because without it, you don't concentrate testosterone in the Sertoli cell. Luteinizing hormone is required by the Leydig cells to make testosterone. And follicle stimulating hormone is very necessary for the initiation of spermatogenesis. Work in laboratory animals suggests it's not necessary for its maintenance. This shows you the phagocytosis of the residual bodies by the Sertoli cells. Interestingly, any of the molecules made by Sertoli cells required for germ cell maintenance are encoded on the X chromosome. And when you have a mature sperm, it has a bullet-shaped head with an acrosome at its tip. It has a midpiece below the head that is very, 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 very rich in mitochondria. And then a tailpiece that is a flagellum that consists of a 9+2 microtubule-driven flagellum that whips side to side, back and forth, and propels the sperm in the direction it needs to go. So the seminiferous tubules are drained by the testes, which are drained by the efferent ductules, which are drained into the epididymis. And you can see this here. All right, so the sperm enter the epididymis through the head region. They're stored there usually for a few months. In the epididymis, the sperm mature. They acquire motility in the epididymis, and they wait patiently, so to speak, for ejaculation. So this would be the head of the sperm right here. Sorry, head of the epididymis. This would be the tail of the epididymis here. During ejaculation, now keep in mind the seminiferous tubules are surrounded by smooth muscle, and those contract via peristaltic contractions. That's how the sperm move from the seminiferous tubules to the root testes, to the efferent ductules, and so on and so forth. So they travel into the vas deferens that you can see here. These are two long tubules that are part of the so-called spermatic cord. That would be the spermatic cord right here. They enter the pelvic cavity through the superficial inguinal ring. The left and the right vas deferens, they go behind the bladder. They descend along the posterior bladder wall. That's what I don't like about this picture here. It gives you the impression that these go through the bladder. They don't. They're on the surface of the bladder. They dilate in a region called the ampulla. Right here, the ampulla. Before narrowing again as they continue through the so-called ejaculatory duct. The ejaculatory duct. Through the ejaculatory duct, they're going to empty into the urethra. Notice now they come into contact with some excretory sex glands. So the seminal vesicles lie adjacent and left to the right ampulla, and they make seminal fluid, which is loaded with fructose. This is an important energy source for the, so the seminal vesicle will load seminal fluid loaded with fructose into the sperm. Then they empty into the urethra, going through this walnut-shaped, walnut-sized gland called the prostate that wraps around the urethra and makes prostatic fluid. Beneath the prostate, there's a pair of bulbourethral glands, and they make a thick, clear mucus that acts as a lubricant. Together, these secretions nourish the sperm, protect them from the acidic vaginal environment, giving them a better chance at reaching and fertilizing an oocyte. These fluids together form what we call semen. Semen, sperm plus the secretions of the three accessory sex glands, the seminal vesicle, the prostate, and the bulbourethral glands. This is ejaculated through the urethra. The urethra is also the exit point for urine. And so the urethra is contained here in what is sometimes called the penile urethra. The penile urethra. So this part sometimes gets called the prostatic urethra. And this is called the penile urethra sometimes. So let's talk a bit about the epididymis. The epididymis is a tightly coiled structure. If you stretched it out, it'd be about 5 meters long, and it's drained by the vas deferens. The head of the epididymis, the spermatozoa are non-motile, probably due to the low pH of the fluid in the epididymis. Modifications of the epididymis allow them to become more motile, more resistant to changes in temperature and pH. Here are some cross sections of the vas deferens. Notice it's not just an empty tube. It's a highly muscular tubule that will contract and powerfully contract and bring the sperm into the urethra. Notice this is lined with a pseudo-stratified columnar epithelium. Here's a basal population that acts as a stem cell population to replace these cells. They sit on a proper basement membrane. Then there's lots and lots of smooth muscle. The seminal vesicle, once it gets to the ejaculatory duct, which is usually closed, it's only open during ejaculation. Seminal vesicle puts in a fructose-containing solution. There's a seminal vesicle. Here is a slide of the seminal vesicle with its beautiful secretory columnar epithelia. It has a lumen. Notice the lumen is lined with smooth muscle. So this contracts and exits material. From the ejaculatory duct, the mixture is expelled in the prostatic urethra. The prostatic secretions contain citrate, an important buffer, calcium, and coagulation proteins that will help the sperm clump when they get outside the body. And citrate's a very important buffer. Sperm is therefore alkaline because of the citrate. And these are some gee whiz numbers. Volume of ejaculate will be anywhere from 1.5 to 5 mils. Sperm count can be anywhere from 40 to 250 million spermatozoa per mil. Percentage of motile forms, usually about 70%. That goes down after 3 hours to about 60. There's usually some leukocytes. Even having a cold can put more leukocytes into your semen. pH is on the alkaline side of things, but not too alkaline. And then the fructose concentration is anywhere from 100, 150 to about, this says 600, probably more like 550, 570 mg per deciliter. And out it goes through the penile urethra. Here is a cross-section of the prostate. Note the tubal alveolar glands that fill with the citrate-rich solution secreted by the epithelium. Notice too the fibromuscular stroma, and this acts like a sponge. It contracts during ejaculation, releasing its material into the prostatic urethra. And these structures are androgen dependent. Withdrawal of androgen will cause atrophy of these glands. So the male sexual response can be categorized by 3 words. They all begin with E: erection, emission, and ejaculation. So erection is characterized by engorgement of erectile tissues. This increases the length and the width of the penis. And let's now talk about these erectile tissues. So we have the corpora cavernosa on the dorsal side of the penis, and the corpus spongiosum on the ventral side. The corpora spongiosa has an internal part called the bulb, through which the urethra enters the penis. The corpora cavernosa makes its way all the way to the pelvic bones. And you can see this here. Here's your dorsal artery. And your dorsal artery, you can see here. There's your urethra. This is your corpora cavernosa on the dorsal side. And here is your corpus spongiosum. So there's one corpus spongiosum. The corpora cavernosa are paired. And the deep artery of the penis is straight through the corpora cavernosa. Notice that these structures are they're embedded in the perineum here. The urethra runs through the ventral side, and you can see that here. It runs right through the corpus spongiosum. So this erectile tissue will run the whole length of the penis. Erection is achieved by parasympathetic nerve-induced vasodilation of arterioles that go through the length of the penis and allow blood to flow into the corpora cavernosa, the neurotransmitter that mediates this is nitric oxide. Nitric oxide induces the activity of guanosine cyclase, sometimes called guanylyl cyclase, that produces cyclic GMP from GTP. And cyclic GMP will prevent phosphorylation of myosin light chain. So it induces relaxation of the smooth muscle that surrounds the blood vessels, causing vasodilation and engorgement of the erectile tissue. So during sexual arousal, smooth muscle cells relax, more blood flows inside the vascular spaces, the corpora cavernosa distends, they press against the inextensible tunica albuginea that compasses the veins in the penis, therefore blood is unable to drain away. The corpus spongiosum has a more flexible tunica albuginea, so it's less turgid, allowing compression of the penile urethra. So blood comes in but doesn't leave. That causes local engorgement and causing a so-called erection. And this is known as penile tumescence. Penile tumescence. T-U-M-E-S-C-E-N-C-E. Penile tumescence. And you can see your parasympathetic neurons releasing acetylcholine stimulates nitric oxide synthase, which is made from the amino acid arginine, which then makes nitric oxide, which causes relaxation of vascular smooth muscle, causing engorgement. Now, cyclic GMP is degraded by phosphodiesterase isozyme 5. Inhibitors of phosphodiesterase isozyme 5 prolongs the half-life of cyclic GMP and increases the time of the erection. This is the basis of the designing of small molecules to inhibit cyclic GMP phosphodiesterase. So sildenafil and vardenafil, tadalafil all inhibit phosphodiesterase 5, maintains a corporeal muscle relaxation of penile erection. I think it's easy to snicker at the development of these drugs, largely because Pfizer stock shot up after the marketing of Viagra. But these drugs are also used in children with pulmonary hypertension. So yes, these drugs were marketed as, excuse the expression, whoopee pills, but they also have other uses that can be used to save lives in infants with cardiovascular abnormalities. So emission refers to the movement of semen into the urethra due to the osmotic contraction of the vas deferens. Ejaculation is the forcible expulsion of semen from the urethra to the outside through the spasmodic contraction of the vas deferens, the muscles in the urethra, and so on and so forth. That's ejaculation. Emission and ejaculation are stimulated by the sympathetic nervous system. They cause the spasmodic peristaltic contractions of the tubular system, also contractions of the seminal vesicles and prostate, and the contractions of the muscles at the base of the penis. Conscious thought is required for erection, but sensory stimulation of the penis can activate the sacral region without conscious thought. However, conscious thought is extremely important, and I think that's why Scripture constantly tells us to guard our minds. The most powerful sex organ you have is not between your legs, it's between your ears. Guys, remember that. And please, yes, I'm preaching a little bit, but listen up, it's okay. Don't say, oh, when I get married, I'll be fine. No. Guess what? When you wake up the next day after your wedding night, you're the same person. So the control you exercise, the discipline you exercise now will carry over into your married life. So start exercising, developing that discipline now, because you'll have a better married life. Okay, end of sermon. Now fertility. So normal sperm counts will range from, the book says 60. Some fertility specialists will tell me 40. So it's 40 to 60 to 150 million sperm per milliliter of ejaculate. If you have a sperm count below 20, then you're deemed subfertile. That's oligospermia. Oligospermia can be caused by heat temporarily, saunas, hot tubs, things like that. Even hot showers or a fever can do it. Certain drugs can cause oligospermia. Anabolic steroid use, because they decrease endogenous testosterone production and endogenous gonadotropin production, can cause oligospermia. Illicit drug use, marijuana, cocaine, opioids, can also decrease sperm production. So can heavy metal poisoning and what have you, can cause problems with male infertility. Popular procedure after you've had their children is a vasectomy. This is an in-office procedure. The doctor gives the male a shot of local anesthetic, usually lidocaine. You make a small incision in the testes. You snip the vas deferens. You suture them shut, and then you suture the incision in the testes. There is some leakage of sperm, therefore 20% of all men who have vasectomies will have antibodies against sperm. That's why reversing a vasectomy can be tougher than it looks. Male homosexuals will also have antibodies against sperm. So will women who engage in anal intercourse. And these can affect fertility as well. Okay, any questions about the male reproductive system? I have 3 minutes left, so I can just start. The female system. And my computer's freaking out. Try that again. There, that's better. All right, so the female reproductive system consists of internal and external organs that are involved with the process of reproduction. So the internal sex organs are the ovaries, which are the female gonads, the fallopian tubes. These are two muscular tubes that connect the ovaries to the uterus. And the uterus is a strong muscular sac that nourishes the embryo and then fetus that develops inside it. The neck of the uterus is called the cervix. And then the cervix protrudes into the vagina, what is known as a birth canal. And then at the genital opening, you have a series of folds that are known as lips or labia. So the uterus has 3 layers. There's an outer layer that consists of connective tissue, perimetrium. Beneath that, there's a layer of muscle called the myometrium. And then the innermost layer is a stratified squamous epithelium called the endometrium. And we are out of time. So we'll pick this up on Monday, talk about the female reproductive system, and hopefully try to get into labor and delivery, because that's quite interesting. And then that'll be it. All right. So have a great weekend. Are there any questions from anybody?
Just to double check for our final next week, it's over urinary, digestive, reproductive, and 2 other ones, right? So
Respiratory.
So 5 chapters in total.
Yeah, respiratory, and cardiac output was not on the last test, right?
Right.
No. So cardiac output, respiratory, digestive, urinary, reproductive.
Okay, so 5.
Yeah.
Thank you.
Okay, anything else? All right, if there's nothing else, I will see you guys on Monday.